Yes.
All right. So my lab is interested in how a really complex organ like the brain has enough specificity to get all of the wiring correct. So there's about 10 to the 11th neurons in the human brain, and each neuron can make up to 1,000 synapses. So it's estimated there's about 10 to the 14th synapses. And we assume that these are very specific connections, similar to how wires in a house have to be connected correctly. If neurons aren't connected correctly, the lights don't come on. So the question is, how does that happen? Particularly only using a toolkit of 20,000 genes. So this is orders of magnitude fewer genes than we would need to wire up the brain if everything was done on a single gene basis. I study the fruit fly brain, which is a much simpler brain. So there's about 100,000 neurons and 1,000,000 synapses in this brain, but you can see that it still has quite a bit of morphological complexity and about the same number of genes. So clearly it's not the gene number that's dictating how the specificity is generated. So what has been shown is that processes such as alternative splicing and proteins that have different functions in different cells can contribute to this process. I'm going to talk today about age-specific functions, so reusing a developmental molecule later for other functions, and combinatorial interactions between neighboring proteins on the same cell are also really important. My lab's been focusing on a gene called DSCAM2. DSCAM stands for Down syndrome cell adhesion molecule. It was originally identified on human chromosome 21 within a region that's critical for Down syndrome. These proteins are conserved from lower vertebrates all up into humans. There's 2 human genes, there's 4 fly genes, and so far all of them have been shown to mediate homophilic binding. So these are expressed on the surface of neurons and have been shown to have important developmental roles in both mice and flies and fish. The DSCAM2 gene is broadly expressed in the developing brain. So this is just a whole brain, a whole mount using a DSCAM2 antibody. It's expressed in all the major neuropils of the fly brain. And what you can see is that it's not expressed in the cell body. So it's localized specifically to dendrites and axons where the synapses are forming. We don't know exactly how many neurons express DSCAM2, but we estimate it's between 25 and 50% of neurons in the brain. And that in itself creates a problem because now you have a homophilic binding protein that's expressed in lots of neurons. So how do you keep those neurons from sticking to each other, or in DSCAM2's case, repelling each other? So that's one of the questions that we've been trying to figure out. And I'm going to summarize the developmental data just because of the short time today. But we found that it has at least 2 different functions during development. It can act as a classical cell adhesion molecule where it's involved in dendritic targeting in the visual system of flies. And then it can also act as a repulsive molecule, which is kind of counterintuitive because this starts out with an adhesion event. But then what happens is that adhesion is broken and the neurons move away from each other. And this has been shown to be important for forming boundaries in the brain and also for a process called synaptic exclusion, where multiple dendrites from the same cell are prevented from being involved in the same synapse through DSCAM2 and DSCAM1 dependent repulsion. So what I'm going to talk about today is whether DSCAM2 has post-developmental functions, and it does. So we recently published a paper showing the data that I'm going to summarize over the next couple of slides. But this started out with an observation that DSCAM2 was expressed in the peripheral nervous system. So we'd always kind of worked in the brain. And I had a student come into the lab that was interested in doing some electrophysiology at the neuromuscular junction. And he was able to show that one of the two DSCAM2 isoforms. I haven't talked about the alternative splicing, but there's an isoform B and an isoform A, and these mediate isoform-specific homophilic binding. So isoform B is expressed in motor neurons and sensory neurons in the periphery, and it's also expressed in the brain and the ventral nerve cord, you can see here. But isoform A is only expressed in the brain and nerve cord. It's not expressed at all in the periphery. So Lorenzo wanted to look at the neuromuscular junction where the motor neurons are connecting to muscles and see if there were physiological roles for DSCAM2 at this synapse. DSCAM2 is not expressed when these motor neurons are forming their synapses. So we looked during embryonic development, and these images here are just reporters for the two isoforms that we have. So we have GAL4s for isoform A and B. And what you're seeing here is basically background staining in the gut of embryos, but the neurons are not labeled with DSCAM2 when they're actually forming these synapses. It comes on at a later time point, which was kind of our first clue that it might be doing something independent from its normal developmental roles. So to do electrophysiology at the NMJ, you take a third instar larvae that looks like this, and you cut it open, do like an open book preparation, and then you have access to the muscles and to the motor neurons that innervate those muscles. So you can stick an electrode in the muscle and record from that, and you're recording basically from all of the, it's not a single cell recording. It's more of a field potential recording. You're recording all of the activity of the motor neurons that innervate that muscle. And then you can also take the nerve that innervates that muscle and stimulate it. And so that's what Lorenzo did. And he started by looking at spontaneous postsynaptic potential. So just sticking the electrode in the muscle and just recording spontaneous events. These are typically single vesicle release events. And he didn't see a difference in either the frequency or the amplitude of these miniature synaptic potentials. So there didn't seem to be a difference at this level of the physiology of this synapse. But then surprisingly, when he stimulated the nerve and recorded the muscle's response to that stimulation, he found that there was an increase in the amplitude of the response. And this evoked potential is typically what people refer to as synaptic strength. So the strength of the synapse is gauged by the amplitude of this postsynaptic response. And neurons work really hard to keep their amplitude the same. And I'll get into some of the mechanisms for that in a minute. But so it was quite unusual that in the DSCAM2 mutants, we had an increase in this response, or an increase in synaptic strength. When we took those miniature potentials and figured out how many vesicles this represented, we could calculate that in controls, they were releasing about 18 synaptic vesicles, whereas in the DSCAM2 mutants, they're releasing about 30. So you might wonder why a neuron would need to inhibit synaptic strength. I've wondered that myself over the last few years. But I think one easy way to think about it is that synaptic potentiation is an integral part of Hebbian plasticity. So as neurons get activated, they get potentiated, and this can be repeated. And so just in terms of resources and things, there's probably not an infinite amount of potentiation that can happen in a neuron. So therefore, you probably need mechanisms to buffer or inhibit the amount of the strength of the synapse as neurons experience different things. And this has been studied quite a lot. There have been a couple different processes that are known for modulating synaptic strength. one of the functions of sleep is thought to be a process called synaptic scaling, right? And so this has been shown quite well in mice, in many organisms actually, but recently it was shown very well at the EM level in mice that after sleep, synapses get downscaled. So if this represents the weight of synapse A of 10, and this is synapse B that has a weight of 2, what sleep will do is downscale everything, but keep the relative differences between these. So A could go to 5 and B could go to 1. So now it has more room to get stronger again, but the relative differences are maintained the same. And that's the process of synaptic scaling. How that works is still being figured out. And then there's also homeostatic plasticity. So I already mentioned that neurons try really hard to keep their synaptic strength the same. How they know what that synaptic strength is supposed to be, we don't really know. But in conditions of disease, synaptic strength can change. And then through homeostatic processes, the neuron tries to bring that back to what it's supposed to be. And this can be through presynaptic mechanisms, such as dumping more synaptic vesicles at the synapse, or postsynaptic mechanisms by recruiting more postsynaptic receptors. And both of those can help increase the amplitude of the response. Okay, so the bottom line then was that DSCAM2 seems to be inhibiting synaptic strength, and we wanted to know, is it DSCAM2 in the motor neurons, or is it DSCAM2 in the muscle? Because remember, this is a homophilic binding protein. So we used an RNAi to specifically knock it out from different cell types. This is the control, looking at, again, quantal content here. When we knock it down with a pan-neuronal GAL4 or with a motor neuron-specific GAL4, we see an increase in synaptic strength. But when we knock it down with a muscle driver, we don't see an increase in synaptic strength. So from this, we concluded that it's required in the motor neurons, but not the muscles, to inhibit synaptic strength. And this was quite surprising because all of the other functions that we knew of DSCAM2 were dependent on homophilic binding. But if the muscle isn't expressing, we can tell that the muscle's not expressing it, but also we can knock it down in the muscle and nothing happens, which means that it's not functioning there, even if it was expressed at low levels. This suggests a different mechanism through which DSCAM2 is working to inhibit synaptic strength. There are caveats to this because we have a hard time detecting it at the motor neuron terminal. So it's possible that it's acting through the dendrites and signaling to the synapse for all of these things, but it would be quite unusual that all of these presynaptic changes that we're seeing are coming actually from the dendrites, but that still needs to be worked out. So what we did was we tried to figure out what is the mechanism that leads to this increased synaptic strength in the DSCAM2 mutants. And I'm not going to go through all of the data, but we ended up doing some electron microscopy and looking at the active zones of the mutant motor neurons. And what we found was that there was an increased number of synaptic vesicles at these active zones. So in flies, you have these electron-dense T-bar structures that mark the active zones. These are docking sites for synaptic vesicles. When we looked at the total number of synaptic vesicles in the motor neurons, they were not different between the controls and DSCAM2 mutants, but the distribution of those vesicles, when we looked at 200 microns around the active zone, we saw an increase in the DSCAM2 mutant synapses. So this suggested a mechanism for how we're getting an increased synaptic strength. There's more vesicles there that can dock and fuse after they receive an action potential. We then went on to try to characterize the signaling pathway that is involved in this process. And we identified a gene called centaurin gamma, which is a PI3 kinase enhancer that was downstream of DSCAM2 or in the DSCAM2 pathway. And synaptic gamma is known for helping to activate PI3 kinase. So this allowed us to draw this signaling pathway here where through DSCAM2 you activate PI3 kinase, and that somehow inhibits the deposition of synaptic vesicles at active zones and thus suppresses synaptic strength. I'm not going to go through all of the data, but we have shown that in the DSCAM2 mutant motor neurons, we have changes in phosphoinositides that you would expect from having a reduced amount of PI3 kinase in there. So we're confident that this pathway is part of the process that we're studying. So that's all the preliminary or the published data that I'm going to talk about. So now I want to talk about what we've been doing more recently. And I've been interested in whether DSCAM2 was involved in learning and memory since I've been studying this protein, which is a really long time now, sorry to say. But I finally had a good reason to look at this, I guess. And so this work started a few years ago with an honors student that I convinced to develop a learning and memory assay in the lab based on, like, we didn't develop this from scratch, other people had developed it previously, but it's always hard to get behavioral assays going. And Kristen Park did that successfully. So this is the assay that we use. It's called a courtship conditioning assay. And it's based on the natural mating behavior of male flies. So males are born with the ability to court females. They know how to do this pretty much as soon as they eclose from their pupal cases. And so what we do is we stick a male that has never seen a female before with a female that's already been mated. And females can store sperm for up to a week, and they don't like to mate again once they've mated. So they express behaviors that are rejective towards the male, and the male will try to court the female. They stick out their wing and sing a song. They have a bunch of different behaviors that we can actually capture on video and then quantify how much time the male spends trying to court the female. So then we take that same male and put him in a different arena with a different pre-mated female and ask how much time he spends trying to court her. If he's learned that females don't like him, he should court less. And that's the learning that we're measuring. So here's an example of one of those assays. We have two fly pairings here, and this male up here is actively courting. You can see him extending his wing and kind of looking at her in the face, and then he chases her around. Whereas this other pair over here are kind of, there's no identifiable courtship behavior here. So we'd be recording this as time spent courting, whereas this behavior would not be recorded as that. And we express this as a courtship index, which is really just the percentage of time that they spend courting in either the training or the testing session. When we do that in wild type flies, we see that in the testing session, there's a decrease in the time they spent courting. And this is the learning that I'm talking about. The males have learned that the females don't like him, and so they court less. When we do that with the DSCAM2 mutants, they don't seem to have a short-term memory at all. So the testing session is equal to the training session. So this was really exciting. The DSCAM2 mutants have a short-term memory defect, and we wanted to explore this further. To try to say that this memory defect was in this pathway that we've been studying in motor neurons, which involves synaptic gamma, we looked at the synaptic gamma mutants to see if they had a memory defect, and we found the same things in the DSCAM2 mutants. So again, they can't form a short-term memory, and this suggested to us that it's this pathway that seems to be involved with short-term memory in the DSCAM2 mutants. So we next wanted to ask, is this role for DSCAM2 a developmental role? Are the neurons screwed up because there's a wiring defect and that's why they can't learn? Or is it a post-developmental role that involves the modulation of synaptic strength? So to do this, we needed a conditional system. So there's a conditional GAL4 system called gene switch GAL4, where the GAL4 won't bind to its UAS element except in the presence of this drug. It's RU486. And so in the presence of RU486, you'll get expression of what's downstream of the UAS. And in this case, it's a RNAi against DSCAM2, so that we can knock DSCAM2 down in a conditional manner. So this is what Josh Davies did in my lab for his honors project this past year. And if you looked at animals that did not contain the gene switch GAL4, they just contained the UAS element, and treated them with drug throughout development, he could see learning. So this was one of the negative controls. When he took a fly that had both the gene switch GAL4 and the UAS RNAi and treated them with the drug throughout development, there was no significant difference in the learning, although there's clearly a trend here. So we're still trying to figure out why that is, why there is this trend towards learning. And when he fed the drug to flies for 3 days after they had closed from their pupal cases, he found that there was, again, a short-term memory defect. So this suggested that it was a post-developmental requirement for DSCAM2, which is really exciting. So we have lots to work on now. So the model that we're working with right now, which could certainly be wrong, but it's kind of the simplest one, so that's what we're going for, is that DSCAM2 is activating the PI3 kinase pathway, which is preventing the deposition of synaptic vesicles at active zones. When you don't have DSCAM2, you have a reduction in PI3 kinase, you get more synaptic vesicles at active zones, and this depletes critical resources for neurons when they need to be potentiated. We don't know if this process is happening. This could be some part of synaptic downscaling that happens during sleep. It could be part of synaptic homeostasis. So DSCAM2 could be one of the players in that whole process. So these are the types of things that we need to figure out. And kind of things that are on our immediate to-do list are to, so if this model is correct and it's really just that they're running out of resources during the day, then we might expect that they would learn better in the morning than they do in the evening. So that's an easy experiment to do to kind of test whether this simple model is correct or not. And then we've also started a series of rescue experiments, one of them using PI3 kinase to see if we can rescue these memory defects by increasing the levels of PI3 kinase in neurons. So I'll leave it at that and just thank the people that did the work. So 3 honors students contributed to this: Kristen Park, Nick Potter, and Josh Davies. And Lorenzo was the PhD student who did all the electrophysiology that I showed you. So thanks, and I'm happy to take questions.
Thank you very much, Sean. That was a lovely talk. I'll perhaps start. We've got a couple on questions on the chat, but I'll start. So you've got this process, this mechanistic difference developmentally versus post-development. What do you think is driving that difference? Is it the post-developmental expression of the SENG1 that's creating this new mechanistic role for DSCAM, or what do you think underlies that?
At this point, I don't know. One thing that we know from motor neurons is that this seems to be an isoform specific process. So isoform B drives this in motor neurons, and isoform A can't do that. We've done this by preventing the neurons from expressing isoform B. So if we make flies where we've knocked isoform A into the endogenous locus, and every neuron that expresses DSCAM2 is expressing isoform A, the motor neurons have a null phenotype. Whereas if we do the same thing with isoform B, the motor neurons look like controls. So it seems to be an isoform B specific thing. And it could be that there's an alternative ligand that is binding to isoform B versus A, but that's one of the kind of burning questions that we want to try to figure out.
Lisa, did you have a question? No. Okay, there's a couple on the chat. Are you able to pull the chat box up, or do you want me to read them out, Sean?
I can do it. Let me just stop sharing my screen.
So Louise does have a question. I'm happy to speak on her behalf, but Louise, don't hesitate, as well as Patricia, to enable your video to come in. But for Louise's question, is PI3K affecting the number of synaptic vesicles by its growth roles? Can you respond to that? Thank you.
By its growth roles? Probably not, because PI3 kinase in neurons is, neurons aren't dividing. They could still be growing, I guess, but they're not dividing. So we think this is through a synaptic vesicle recycling pathway. So data that I didn't show you suggests that there's a reduction in PI3P, which is a critical phosphoinositide for fusing pre-endosomes to endosomes. And so we think that there might be a delay in that process, and that's how you basically recycle your synaptic vesicles. Why that leads to the more synaptic vesicles being deposited at active zones, we don't know. We're trying to figure that out.
Thanks, Louise. If that responds to your follow-up question, that would be great. Otherwise, just enable your video and then you can follow it. But we'll just pass the next question to Patricia, if we could. Thank you.
Sean, very nice talk. In your last model, you talk about resource depletion. Are you talking about running out of synaptic vesicles, basically, in that scenario, because you're releasing?
Yeah.
Can you demonstrate that in the motor neuron muscle assay by repeat stimulation that you don't get that activation?
Do like a run out experiment where we try to deplete them in the DSCAM2 mutant versus controls. Yeah, so we haven't done that, and Lorenzo has a very good reason why he hasn't done that, but I can't remember what it is. But there's some reason that he didn't think we'd be able to interpret it. We have talked about it, but he didn't think it was going to work that well. Yeah.
Mike, after you, Mike.
There you go.
Oh yeah, Sean, I have a question, and we're certainly not big mice, but we're definitely not big flies. But is there anything we can understand from DSCAM2 that we can apply to our understanding of synaptic vesicle loading in human neurons and these behavioral traits that you are observing in these mutant mice with regard to sleep, mating behaviors, and so on? And so we know very well that alterations in DSCAM dosage in Down syndrome is a predominant feature in individuals with Down syndrome and these disability traits. So is there anything that we can understand about the homophilic and the adhesion properties and the Centaurin gamma A signaling pathway that you could identify from the information that's in these mammalian models that can guide you or at least provide some perspective for your Drosophila studies?
Yeah, I mean, we certainly hope so. It's interesting. So the protein domain structure of human and fly DSCAM are identical, right? It's got the same number of Ig domains and fibronectin domains. It looks the same. The cytoplasmic domains of all of the fly DSCAMs and vertebrate DSCAMs are very different, but surprisingly, the signaling pathways seem to be conserved. So, for example, there's PAC and DOC are involved in DSCAM1 signaling in flies, and that's also been shown to be true in vertebrates. And recently, there was a paper that showed that the cytoplasmic tail of DSCAM proteins can actually get cleaved and go to the nucleus and regulate gene expression. That's been shown with both fly and human DSCAM. So I think there are good links there. They just haven't been fully explored yet.
That's really interesting. I didn't realize about that recent work on C-terminal gene regulation. Thank you very much.
And the DSCAM2 C-terminal domain is really interesting as well. So it's got 4 different isoforms, and there are these long isoforms that contain prion-like domains. And prion-like domains have been shown recently to be involved in liquid phase separation. So we're thinking that maybe this is acting as a nucleating molecule at synapses that could help with some of these signaling mechanisms.